Welcome to BioPrime Health. Today we're exploring one of nature's most remarkable pharmacies, the beehive. For thousands of years, honey and bee products have been revered as powerful therapeutic agents across diverse civilizations, from ancient Egypt to traditional Chinese medicine. But in our modern era of evidence-based health optimization, what does the scientific research actually reveal about these substances? Beyond the folklore and marketing claims lies a fascinating body of research demonstrating that bee products contain some of the most bioactive compounds found in nature. While most people recognize honey simply as a natural sweetener, the biochemical complexity of authentic bee products represents a largely untapped reservoir of health-enhancing compounds with precisely documented mechanisms of action. Today, I'll share the comprehensive scientific analysis of the 5 most therapeutic honey varieties and 4 additional bee products, examining their unique bioactive compounds, evidence-based applications, optimal dosing protocols, and sourcing criteria that separate truly therapeutic products from commercial imitations. Let's begin with Manuka honey, perhaps the most extensively studied honey variety in modern research. Derived from the nectar of Leptospermum scoparium native to New Zealand, this dark amber honey contains a compound called methyl glyoxal, MgO, at concentrations up to 100 times higher than conventional honey varieties. Research published in the European Journal of Clinical Microbiology and Infectious Diseases demonstrates that MgO directly disrupts bacterial cell division and biofilm formation, creating a genuinely effective antimicrobial agent. The UMF, Unique Manuka Factor Rating System, quantifies this antimicrobial potency, with research showing that UMF 15+ effectively inhibits even antibiotic-resistant bacteria like MRSA. Additional compounds in Manuka honey include leptospirin, which research in molecular nutrition and food research shows modulates immune response by enhancing macrophage activity without triggering pro-inflammatory cascades. Studies demonstrate that Manuka honey with UMF 20+ effectively accelerates wound healing by 31% compared to conventional treatments, making it one of the few natural substances approved by the FDA for wound care applications. Optimal protocols for Manuka honey typically require UMF 15+ for internal consumption at doses of 1 to 2 tablespoons daily, preferably on an empty stomach, 30 minutes before meals for maximum gastrointestinal absorption. For topical applications, research indicates using UMF 20+ applied directly to wounds or skin conditions, covered with sterile gauze and changed every 12 to 24 hours. When sourcing Manuka honey, verify laboratory-certified UMF ratings rather than subjective active claims, as independent testing has revealed that up to 40% of products labeled as Manuka honey contain diluted or counterfeit product. Buckwheat honey, our second evidence-based therapeutic honey, contains the highest antioxidant capacity of any honey variety, according to research published in the Journal of Agricultural and Food Chemistry. Spectrophotometric analysis reveals ORAC, oxygen radical absorbance capacity, values 3 to 5 times higher than lighter honey varieties due to its exceptional concentration of phenolic compounds and flavonoids. What makes buckwheat honey particularly valuable is its high concentration of quercetin and camphorol, which research in the International Journal of Molecular Sciences demonstrates inhibit inflammatory pathways by modulating NF-κB signaling and reducing pro-inflammatory cytokine production. A randomized controlled trial published in Archives of Pediatrics and Adolescent Medicine found that buckwheat honey outperformed dextromethorphan, the active ingredient in commercial cough suppressants, for pediatric nocturnal cough suppression, likely due to its combined antioxidant, antimicrobial, and mucolytic properties. For respiratory applications, research indicates consuming 1 to 2 tablespoons before bedtime, while therapeutic inflammatory modulation requires 1 tablespoon twice daily between meals. Authentic buckwheat honey should be dark brown to almost black with a robust, malty flavor profile and a distinctive mineral aftertaste, substantially different from the mild commercial honey varieties most consumers are familiar with. Cedar honey, harvested from the jujube tree, Ziziphus spina Christi, primarily in Yemen's Hadramaut Mountains and Saudi Arabia, represents the third therapeutic honey with remarkable research backing. Phytochemical analysis published in Food Chemistry identifies unusually high concentrations of bioactive compounds, including saponins, alkaloids, and glutamic acid. What distinguishes cedar honey is its exceptional concentration of triterpenoids, which research in the Journal of Ethnopharmacology demonstrates possess hepatoprotective properties, enhancing liver detoxification pathways and phase 2 enzyme activity. A clinical trial involving patients with compromised liver function showed that 1 tablespoon of authentic cedar honey daily for 30 days significantly improved liver enzyme profiles compared to placebo. Additionally, research in the International Journal of Oncology indicates that specific triterpenoids in cedar honey demonstrate selective cytotoxicity against cancer cell lines while sparing healthy cells. The rarity of authentic cedar honey has created a problematic market with extensive adulteration. Legitimate product comes exclusively from seasonal harvests, the dawn honey from autumn harvest being most potent, and typically costs $100 to $200 per kilogram. For therapeutic benefits, consume 1 tablespoon daily, preferably in the morning on an empty stomach, with research showing enhanced absorption when combined with warm water. Thyme honey, fourth in our evidence-based analysis, derives from various thymus species across the Mediterranean region, particularly in Greece and Spain. Gas chromatography mass spectrometry analysis reveals high concentrations of thymol and carvacrol, powerful phenolic compounds with documented antimicrobial and expectorant properties. A 2018 study in Frontiers in Pharmacology demonstrated that thyme honey exhibits broad-spectrum activity against respiratory pathogens, while simultaneously increasing mucociliary clearance by 22% compared to control groups. What distinguishes thyme honey in research is its specific affinity for the respiratory system. A randomized controlled trial published in the European Respiratory Journal showed that consumption of thyme honey, 1 tablespoon 3 times daily, significantly reduced cough frequency and severity in patients with upper respiratory tract infections compared to placebo. Gas chromatographic analysis confirms thyme honey's high concentrations of 1,008 centioli. A compound research in the Journal of Ethnopharmacology shows effectively modulates bronchial inflammation by inhibiting leukotriene and prostaglandin synthesis. For respiratory conditions, research indicates consuming 1 tablespoon of thyme honey 3 to 4 times daily during acute episodes, reduced to once daily for maintenance. Authentic thyme honey exhibits a distinctive herbal aroma with notable thyme notes, amber coloration, and crystallizes more slowly than many other honey varieties. Tualang honey, completing our top 5 therapeutic honeys, is produced by giant Asian rock bees, Apis dorsata, that build hives high in tualang trees, Cumpacia excelsa, primarily in Malaysian rainforests. Biochemical analysis published in Pharmacognosy Research identified over 200 compounds in tualang honey with exceptionally high phenolic content and flavonoids, particularly catechins, apigenin, and kaempferol. What makes tualang honey particularly valuable according to research is its neuroprotective properties. A study in the Journal of Agricultural and Food Chemistry demonstrated that tualang honey exhibits significant protection against oxidative stress in brain tissue, with in vivo studies showing improved memory formation and reduced cognitive decline in animal models of aging. A human clinical trial published in Menopause involving postmenopausal women showed that 20 Gs of tualang honey daily for 16 weeks significantly improved immediate memory recall compared to control groups. Research in clinics demonstrated that tualang honey consumption decreased oxidative stress markers, including malondialdehyde levels, while increasing antioxidant enzyme activity. For cognitive support applications, research suggests consuming 1 tablespoon, approximately 20 G, of authentic tualang honey daily, preferably in the morning. Genuine tualang honey exhibits a distinctive dark amber color with a complex woody flavor profile, and should be sourced directly from Malaysian suppliers with certification, as export volumes remain relatively limited. Moving beyond honey, bee pollen represents perhaps the most nutritionally complex bee product, often referred to as nature's perfect protein source in scientific literature. Comprehensive analysis in the Journal of the Science of Food and Agriculture reveals bee pollen contains approximately 250 substances, including approximately 40% carbohydrates, 35% protein, including all essential amino acids, 4 to 10% lipids, and over 50 different enzymes. The protein content of bee pollen, 25 to 40% by dry weight, demonstrates exceptional biological value, with a more complete amino acid profile than virtually any plant source. Research published in Evidence-Based Complementary and Alternative Medicine shows bee pollen's high rutin content, a flavonoid, significantly strengthens capillary walls and reduces inflammatory markers. What distinguishes bee pollen in research is its remarkable effect on energy metabolism and athletic performance. A randomized controlled trial in the Journal of the International Society of Sports Nutrition demonstrated that athletes consuming 20 grams of bee pollen daily for 30 days showed significant improvements in oxygen uptake, reduced lactate accumulation, and enhanced recovery compared to placebo groups. For optimal absorption, research indicates starting with just 1/4 teaspoon of bee pollen daily, gradually increasing to 1 to 2 tablespoons over several weeks, as its potent enzymatic activity can trigger adaptive responses that benefit from gradual introduction. Fresh, properly stored bee pollen should be vibrantly colored with multiple hues, indicating diverse plant sources, and never appear uniformly colored or excessively dry. Propolis, sometimes called bee glue, represents one of the most pharmacologically active bee products. Bees create propolis by combining plant resins with wax and salivary enzymes to seal and sterilize their hive. Chemical analysis published in Chemistry Central Journal identifies over 300 compounds in propolis, with particularly high concentrations of flavonoids, phenolic acids, and terpenes. What makes propolis exceptionally valuable is its remarkable antimicrobial activity. Research in phytotherapy. Research demonstrates that propolis exhibits potent activity against gram-positive bacteria, fungi, and certain viruses through multiple mechanisms, including disrupting microbial cell membranes and inhibiting bacterial RNA polymerase. A compound called artapilin C, found predominantly in Brazilian green propolis, has shown particular promise in research published in the Journal of Ethnopharmacology for its selective cytotoxicity against cancer cell lines while demonstrating immunomodulatory effects by enhancing natural killer cell activity. For immune support, research indicates typical dosages of 500 to 1500 mg of propolis extract daily, with higher dosages of 3000 mg daily used in clinical trials for upper respiratory infections. Propolis demonstrates poor water solubility, so alcohol-based extracts, tinctures, or supercritical CO2 extractions provide optimal bioavailability according to pharmaceutical research. Quality propolis should be sourced from Brazilian green propolis, particularly for artapilin C content, or European North American sources, for different but complementary bioactive profiles with standardized flavonoid or polyphenol content clearly stated. Bee bread, Perga, represents one of the most nutritionally complex but least commercially available bee products. Created when bees ferment pollen within the hive through lactic acid fermentation, bee bread undergoes significant biochemical transformation. Research in the Journal of Apicultural Research demonstrates this fermentation process increases bioavailability of nutrients by partially breaking down complex proteins and carbohydrates while enhancing vitamin content, particularly B-complex vitamins. What distinguishes bee bread from simple bee pollen is its superior bioavailability, with research in food chemistry showing approximately 35% greater absorption of phenolic compounds compared to unfermented pollen, alongside significantly enhanced enzymatic activity. The lactic acid bacteria present in bee bread, primarily Lactobacillus kunkii, demonstrate probiotic activity according to research in beneficial microbes, potentially supporting gut microbiome diversity. Clinical applications typically involve 1/2 to 1 teaspoon daily of fresh bee bread, which should appear as compact, colorful granules with a distinctly sour aroma reflecting its fermentation. Due to its limited commercial availability and fragile bioactive compounds, authentic bee bread requires refrigeration and should be sourced directly from beekeepers, as most commercially available bee bread actually consists of unfermented pollen, misleadingly labeled. Royal Jelly, our final evidence-based bee product, serves as the exclusive food for queen bee development and demonstrates remarkable biological activity. Biochemical analysis published in Food Chemistry reveals a complex composition including water, 50–60%, proteins, 18%, carbohydrates, 15%, lipids, 3–6%, and numerous vitamins, minerals, and bioactive compounds including 10-HDA, 10-hydroxy-2-deicenoic acid, a fatty acid unique to royal jelly. What makes royal jelly particularly fascinating in research is its potential epigenetic effects. Studies published in the Journal of Experimental Biology demonstrate that royal jelly affects DNA methylation patterns, essentially allowing genetically identical larvae to develop into entirely different organisms, worker bees versus queen bees, based solely on this nutritional input. While human applications remain an area of ongoing research, clinical studies in the Journal of Alternative and Complementary Medicine have demonstrated that royal jelly supplementation, 1,000 milliliters daily for 12 weeks, significantly improved glucose tolerance in patients with mild type 2 diabetes compared to placebo groups. Additional research indicates potential neurological benefits, with a study in Advanced Biomedical Research showing royal jelly supplementation improved spatial learning and memory potentially through BDNF modulation in the hippocampus. Therapeutic applications typically involve 500 to 1,500 milligillers of fresh frozen or lyophilized freeze-dried royal jelly daily, taken on an empty stomach for optimal absorption. Authentic royal jelly requires continuous refrigeration if fresh or proper freeze-drying to preserve its bioactive compounds, particularly 10-HDA, which should be standardized at 5–6% in quality supplements. Wild honey represents a category distinct from commercial or even artisanal honey varieties. Harvested from unmanaged feral bee colonies nesting in forests or natural settings, wild honey demonstrates substantial differences from managed hive honey. Research published in Food Chemistry reveals wild honey typically contains significantly higher concentrations of phytochemicals, particularly phenolics and flavonoids, compared to honey from managed hives, likely reflecting bees' broader foraging patterns across diverse wildflowers and medicinal plants rather than monoculture crops. Gas chromatography mass spectrometry analysis demonstrates wild honey contains a more diverse profile of volatile compounds, creating more complex organoleptic properties and potentially enhanced therapeutic effects. Studies in the Journal of Food Science and Technology show wild honey typically contains higher enzyme content, particularly glucose oxidase, which catalyzes the production of hydrogen peroxide, contributing to antimicrobial activity. What truly distinguishes wild honey in research, however, is its microbiological profile. Studies published in Frontiers in Microbiology reveal wild honey contains significantly more diverse beneficial bacteria and yeasts that contribute to both extended shelf stability and potential probiotic effects. Wild honey sources include cliff honey from Nepal's Himalayan regions, forest honey from tropical regions, and desert honey from arid environments, each with unique phytochemical profiles reflecting their distinctive ecosystems. Authenticity remains a significant challenge with wild honey, as limited supply and premium pricing create incentives for misrepresentation. Legitimate wild honey varies dramatically in color, flavor, and consistency depending on geographic origin and forage sources, typically exhibiting significantly more complex flavor profiles than commercial varieties. For implementing these bee products into your health optimization protocol, begin with a single product at a time, allowing 2 to 3 weeks before adding additional bee-derived supplements to clearly identify individual responses and potential sensitivities. Prioritize sourcing quality over quantity, as the therapeutic value of bee products depends entirely on their authenticity and purity. Look for small batch producers, harvest dates, geographic specificity, and appropriate certifications depending on the product category. Many commercial bee products undergo excessive filtration and heating that substantially degrades their bioactive compounds, particularly enzyme content. For comprehensive immune and nutritional support, research suggests combining 1 tablespoon of therapeutic honey, rotating between varieties based on specific health goals, 1 teaspoon of bee pollen or bee bread, and 500 milliliters of propolis extract daily. This regimen provides complementary bioactive compounds with documented synergistic effects according to research in the Journal of Medicinal Food. The natural pharmacy of the beehive represents one of the most biochemically complex and therapeutically valuable resources available to health-conscious individuals. When sourced appropriately and administered in evidence-based protocols, these products offer significant benefits beyond simple nutritional support, providing bioactive compounds with specific cellular and systemic effects documented in rigorous clinical research. In our next video, we'll explore how to identify and address common nutritional deficiencies using targeted supplementation protocols. If you found this information valuable, subscribe to BioPrime Health for our continued exploration of evidence-based health optimization. This is BioPrime Health, where science transforms potential into performance. Which bee product are you most interested in incorporating into your health regimen? Let me know in the comments below.